Estamos ao vivo, pode começar.
Hi, everybody. Good afternoon. It's a great pleasure for me to be here with you today, starting our first Immunolac seminar of the year. And it's a great pleasure to receive two fantastic colleagues from Mexico and Chile to talk with us about our favorite...
Pode começar.
Oh, sorry. Hi, everybody. Good afternoon. It's a great pleasure for me to be here with you today, starting our first, I'm sorry, apologies. I had the YouTube turned on, so it's a little bit delayed. So again, so it's great to be here with you all. And so we have over 400 people registered for the seminar today to watch Dr. Leopoldo and Dr. Karina to give their presentation. So thank you very much for your participation here. And without any further delays, we'll get started with the seminar, which will start with Karina. So I will give a brief introduction about her, so you guys get to know her and a little bit about her research. So Dr. Karina Pino Lagos is an immunologist that attended the University of Chile between the years 1998 and 2005, receiving a professional degree in engineering in molecular biotechnology. Then Karina joined the molecular and cellular biology program at the Dartmouth College in New Hampshire, United States, where she got her PhD degree. And then in 2011, she went back to Chile and worked as assistant professor at the Faculdade de Medicina da Universidad de Chile. And 4 years later, she joined the Faculdade de Medicina at the University of Los Andes in Santiago, where she's now an associate professor and the head of the immune regulation group, which belongs to the program in immunology hosted by the Center of Investigations and Innovations in Biomedical Sciences in the same university. So Karina is interested in investigating the process of immune tolerance, which is of great interest to all of us, of course. And her research centers in understanding the role of a small population of T cells, but quite important in regulating the immune response, which are the regulatory T cells, and also have an important role in the homeostasis in our body. So today, Karina will present to us about the role of neuropilin-1 on regulatory T cells. So I thank Karina very much for being here with us and representing Chile in this series of seminars. Thank you, Karina. Carry on.
Okay. Well, first of all, thank you to the Sociedade Brasileira de Imunologia for the invitation and for the audience, of course. Today, as the presenter just told you, I'm going to talk about our research line, which is involved in the induction or trying to understand how tolerance is established through the function of a small population of T cells called regulatory T cells, and also in a very interesting protein for us called neuropilin-1. This protein, neuropilin-1, is a multifunctional co-receptor that was first described in the nervous system, related to the axonal guidance in neurons. And later on, it was published that neuropilin-1 can be expressed by different immune cells, including dendritic cells and T cells. In a human system, using human cells, of course, but in vitro, was studied and described that neuropilin-1 was required for a stable interaction between dendritic cell and regulatory T cell, and perhaps mediating the function of these regulatory T cells, because its presence allowed this interaction, in contrast to effector cells that do not express elevated levels of this protein. In 2008, 2012, it was described that neuropilin-1 could serve as a potential marker for the identification of regulatory T cells in vivo, because neuropilin-1 expression can be correlated with FOXP3. But now we know that that's not true. It could serve as a marker. I think it depends on the setting or the criteria that you are using. So because for neuropilin-1, it was studied in mucosal immunology first, and we were interested in the induction of immune tolerance, but in the system of experimental system of transplantation. We were wondering if the neuropilin-1 expression follows the same rules that have been described previously in this mucosal immunology setting, both in gut and also in the lung. So we decided to perform the skin transplantation experiments in mouse, as we do in the lab. And in this setting, we take B6 animals, B6 mice that receive a syngeneic or an allogeneic piece of skin from the donors. And we wait for 20 days and decided to analyze what was happening with the expression of neuropilin-1 on T cells, both effectors and also on regulatory T cells in these different settings. So here on top in the panel A, you can see if we get it on CD4 T cells, we get 17% of the CD4s expressing neuropilin-1. But in the allogeneic group or the rejection group, the T cells, all the T cells, were expressing higher levels of neuropilin-1. And this was first, this observation was new at the time because it was described before that effector T cells were not able to express neuropilin-1. So in this setting, under inflammation, we could see that CD4s were increasing neuropilin-1 expression. And then if we used this criteria, as I mentioned earlier, using CD4 as a marker to identify regulatory T cells together with high expression of CD25 and neuropilin-1, we use that criteria to sort regulatory T cells and transfer them together with effector T cells that were not neuropilin-1 positive into RAG knockout mice. And we did the skin transplant using F1 skin or the allogeneic skin. And we found that if you use the neuropilin-1 positivity along with high expression of CD25 as a way to identify these natural Tregs, we can see that those cells are able to allow the acceptance of a skin graft. In this same study, we decided to track the expression of neuropilin-1 on effector cells and also on regulatory T cells. And to do that, we use congenitally marked animals, and we also use FOXP3 reporter mice. And here is the setting, the experimental setting, basically using this congenitally marker to identify these both populations into these mouse that will be able to accept or reject a skin graft. So first, we analyze the expression of neuropilin-1 on the natural Tregs following the expression of CD4s and Li5.1 positive cells. And here you can see that there's no variation on the expression of neuropilin-1, on CD4s, on specifically on Tregs, in the different context, right? Either in the syngeneic group or in the allogeneic group. But the interesting observation was made when we analyzed the effector CD4 positive cells. In that case, what we found was that if we have a mouse that received effector cells that were not expressing neuropilin-1 when we did the injection in the mouse, and we add a syngeneic piece of skin, the expression of neuropilin-1 reached 50% of CD4s, more or less. But in the same group, if you add the injection of the Tregs that we know that are neuropilin-1 positive, the upregulation of neuropilin-1 is 20% more than the condition without the Tregs. But with effectors, both same type of transplant. Meaning that something was happening between the interaction of the Treg and the effector, because the effectors were gaining neuropilin-1 expression in the presence of the Tregs. What happened in the rejection group, the same, but the difference was even higher. So we have a rejection group with effector cells expressing 35% of neuropilin-1, and this frequency increased when the effector cells are co-injected with the regulatory T cells. So with this, in this publication, that was the thesis of one of my undergrad students back in 2014, sorry. We could say that the transfer of the regulatory T cells was mediating the upregulation of neuropilin-1 on effector cells, but we didn't know what was the consequence of this observation, right? Because at that time we didn't have the tools to try to answer that question. So then we could obtain these animals. These are conditional knockout animals that allow us to evaluate the contribution of neuropilin-1 specifically on FOXP3 positive Treg. So we have one line of animals that express FOXP3 with the reporter gene YFP, so allow us to see the Tregs in the mouse, and we could sort them. And the second line that expressed a fluxed neuropilin-1. So when we cross these two mouse lines, we get, after several crosses, not only one, we get mice, the neuropilin-1 knockouts that we call, that are those animals that only regulatory T cells cannot express neuropilin-1. So that way we could ask what was neuropilin-1 really doing on these Tregs, and if that presence will have or not an impact on the effectors. So then we did the experimental, the basic in vitro suppression assay, in which we take effector cells, we stain them with a dye, either CFSE or CTV, and we stimulate them in the plate, and we add different ratios of regulatory T cells from the three different genotypes, the wild types, the heterozygous, and the homozygous for neuropilin-1. And as you can see here in this slide, we did not find any difference in the capacity of regulatory T cells to suppress the proliferation of the effectors. And this setting is a contact-dependent setting, because regulatory T cells and the effectors are in the same well. So maybe we thought perhaps the lack of neuropilin-1 is being compensated by other molecule that could be involved in the suppression mechanism of regulatory T cells. So why don't we repeat this experiment, but in a different setting, which is the contact independent. So we use the transwell plates, and we did exactly the same experiment. In this case, we have the effector cells on the bottom and the Tregs separated from the effectors, the Tregs on top. And what we found here is that, yes, in this case, we do see difference in the suppression activity by the regulatory T cells when the cells are separated, meaning that a soluble factor could be involved in the suppression mechanism of regulatory T cells that is neuropilin-1 dependent. If you remember from the first slides, I showed you that in the in vivo, the very basic or general experiments that we started to do at the very beginning, we observed that when you had these the regulatory T cells together with the effector cells, the effectors were gaining neuropilin-1 expression. So here we checked what was happening with the expression of neuropilin-1 on the effectors when we use the wild-type Tregs and the knockout Tregs for neuropilin-1. And again, we found that the effector cells, when the Treg does not have the ability to express neuropilin-1, the effector cell in the white bar is unable to upregulate neuropilin-1 on their surface as well, okay? Then we move to the in vivo experiments, and the same experiment I explained you before. We have all the different groups. We have rag knockout animals with the F1 or the allogeneic skin. We transfer the conventional or effector T cells that we know, we sort them for CD4 positive, CD25 positive, neuropilin-1 negative, and the CD25 negative and neuropilin-1 negative, and the regulatory T cells from the 3 different genotypes. And following the same results as before, if we transfer wild-type regulatory T cells are allowed to accept an allograft, whereas the regulatory T cells that are not expressing neuropilin-1 do not, or the survival of the skin grafts is reduced. So we know that there is an interaction between these two cells, right? So we wanted to know what was happening with the phenotype. Is perhaps thanks to the interaction between neuropilin-1 on the effectors and also the regulatory T cells, favoring or facilitating certain phenotype on the effectors and the same on the regulatory T cells. And we didn't find much differences on the effectors, but on the Tregs, we did observe that those regulatory T cells that do not express neuropilin-1 and are in the mouse facing a skin graft, that we know those animals will not accept the skin graft, right? These Tregs produce less IL-10 than the wild-type groups. so here is the quantification. So from this experiment, we conclude that neuropilin-1 is required by regulatory T cells to produce IL-10 in the skin transplantation or in the skin acceptance rejection response. Because in the Transwell experiment, we knew, right, that there was a soluble factor that on one side could be IL-10, but on the other side could be other molecules that the regulatory T cells are secreting. And since 2008 is being kind of more known, or I don't want to say popular, but it's being hot lately that even immune cells are able to secrete small extracellular vesicles, and through the secretion of these vesicles, cells, not only immune cells, of course, can communicate and modulate what is happening in the neighborhood, right? So there were a couple of papers saying that, in fact, regulatory T cells can secrete these extracellular vesicles or EVs, and through the release of these vesicles, the Tregs can suppress the immune response. So we wanted to know if in our hands, the Tregs were producing exosomes or extracellular vesicles, and if neuropilin-1 was present on these vesicles as well, if it has any role on the suppression mechanism through the release of these EVs. So we isolated natural Tregs from reporter animals for FOXP3, and these natural Tregs were cultured for 48 hours, and we collected the supernatant. And then after ultracentrifugation, we enriched for small extracellular vesicles that the samples can be checked by the presence of these small EVs by nanotracking analysis, which is an equipment that gives you this type of plot and allows you to visualize the size of the particles that the cells are secreting into the media. And these EVs have a characteristic size of 150 nanometers. So we compare the production, the amount, and the size of the EVs produced by wild-type Tregs, and also for those Tregs that are not expressing neuropilin-1. And we did not find major differences either in the concentration or in the size of the vesicles. We also perform, this is a very, like the standard protocol to characterize the EVs that you are studying, is by doing a Western blot against these two proteins that serve as a marker of EVs. So we could find ALIX and TSG101 on the EVs coming from wild-type Tregs and the knockout. And very important for us, and it was new as well because there were no papers describing the presence of neuropilin-1 on the extracellular vesicles from natural Tregs. So here we can find the protein in the membrane, and in the knockout and the EVs coming from knockout cells, neuropilin-1 is not present. So then we did a suppression assay in vitro. We take effector cells, stain them with a dye, and we add polyclonal activation, anti-CD3, anti-CD28, and then we add the EVs coming from wild-type Tregs or the knockout. And here, if you just focus in these two conditions, this is the wild-type EVs coming from wild-type Tregs, and here the ones from the knockout, and those EVs are not able to suppress the proliferation of the effector T cells. Then in vivo, same thing. We did the in vivo, the setting that we used. Here, different to what most of the publications show, we decided to go local here. In this setting, we didn't inject the EVs intravenously, but subcutaneous around the area where the skin graft was going to take place on the next day. So the day before, or the day -1, we shave the back of the mouse and we draw a square. We do 4 injections of the EVs, and this mouse also receive the effectors. These are rag knockouts. Receive CD4 positive T cells, and then on day 0 we do the surgery, the skin graft, and we found that if you treat these animals with extracellular vesicles coming from wild-type Tregs, not Treg cell, just the EVs, we see around 60% of graft acceptance. But if these EVs come from neuropilin-1 knockout Tregs, the acceptance of the skin grafts is less frequent. It's about 30% on the mice. We decided to check what was happening in the skin graft, thinking that because we did the injections were local on the site of the transplantation. So we took the skin graft and we checked for different antigen presenting cell population. We also included neutrophils, basophils, and others. And the only differences that we found was on the ratio of macrophages, specifically on M2s. So here, we don't see differences on the frequency of M1 type macrophages, but in those animals that receive the EVs from wild-type Tregs, the M2 type of macrophages are more frequent on the skin graft in comparison with the condition in which the animals receive the EVs from the neuropilin-1 knockout Tregs. So from this piece of data, we can tell that these EVs that have neuropilin-1 on the surface are doing something, and that something at least is through the presence of the macrophages in the transplant. And this is just to finish. We had these EVs sent to Germany to the lab of one of our collaborators, and they did the proteomic, and we found the differences in the enrichment of certain proteins. Here you can see in the first 2 columns the heat map for the wild-type EVs or the ones coming from the knockout Tregs. And we found, for example, that the EVs coming from neuropilin-1 knockout Tregs have less CD73, that we know is a immune regulatory related molecule. We also found that it has less CD25, CD82, granzyme B, CD43. This is just what I just mentioned, and we corroborated that CD73 is less abundant on the EVs coming from neuropilin-1 knockout Tregs, doing the Western blot assays, and we are doing currently this verification on CD25 and CD43 as well. So as a conclusion, neuropilin-1 is required by regulatory T cells to suppress T cell proliferation in vitro, to favor allogeneic transplantation in vivo to produce IL-10 and to produce the immune activation via the release of small extracellular vesicles. This is the acknowledgement to my laboratory and also to our collaborators and the funding from our national agency, and again to the Society of Immunology in Brazil, and also the Association for the invitation. Thank you.
Thank you very much, Karina, for a very interesting and exciting presentation. I've got a question here that was sent directly to me from Josiane Silva in University of São Paulo, and she has actually two questions. One, I think you partially responded, which is whether neuropilin-1 also has the ability to induce IL-10 in other cell types, and whether neuropilin-1 has an effect in inducing the expression of other modulatory molecules such as CTLA-4, for example.
Okay, very good question. So, at least from the analysis we did ex vivo from the animals receiving the different type of regulatory T cells, we did not see differences on IL-10 by effectors. Perhaps it's not a good population to look at because under rejection, those effectors are not going to produce much IL-10. But because now we observe that it may have an effect on the macrophages and the M2 type, we should be looking at IL-10 in that population. We hope to do those experiments this year. But what we are doing for sure is we are doing transcriptomic on CD4 T cells, conventional or effector CD4 T cells, with the EVs coming from the wild type or the neuropilin-1 knockout. We saw some, for example, reduction on FOXP3 expression on the effectors and upregulation of interferon gamma and IL-17, but this was just by qPCR. We have not checked at the protein level. And those are the experiments we are starting this year of the grant because it's one of the objectives that we proposed. So we hope to have some data at the end of the year.
Perfect. Thank you very much, Karina, for exciting presentation.
You.
And we also thank your society for indicating you to come speak for us. And I'd like to say that our president from ALACI, Dr. Emilio Malchiodi, is present here. So thank you very much, Emilio, for coming and seeing us. So thank you, Karina. And we are now going to move on to Dr. Leopoldo Santos Argumedo's presentation. Leopoldo is the principal investigator and head of the Department of Molecular Biomedicine at the Center for Research and Advanced Studies of the National Polytechnic Institute in Mexico. He has a degree in microbiology as well as a master's and a PhD, both specializing in immunology. He did his postdoc studies in Mill Hill in London and in the Department of Immunology at the Next Research Institute, where I also used to work for a while long ago. And he has served as the president of the Mexican Society of Immunology and also the Latin American and Caribbean Society for Immunology. So Leopoldo is a very active member of the Latin American immunology community. And his research interest is focused on studying the activation, maturation, and differentiation mechanisms of B lymphocytes in humans and mice. In recent years, he has focused his studies on searching the role of class 1 myosins in the immune response. His lab has been analyzing the molecular mechanisms in human primary immunodeficiency for more than a decade, and he also works on IgA characterization and microbiome in human colostrum. So today, Leopoldo will talk to us about class 1 myosin proteins and the adhesion, trafficking, and homing of lymphocytes. So thank you very much, Leopoldo, for being here with us. It's a great honor to have you, along with Karina, opening our first seminar of the series this year.
Well, thank you very much for, for your kind invitation. And let me share my presentation. I hope it works fine.
Perfect.
Okay.
Yes.
First of all, I would like to thank the Brazilian Society of Immunology for this invitation. It's a very good opportunity to talk about the work that we have been doing during the last 30 years. It's a lot of time. And I will start with this slide. This is a very old slide. Let me tell you my story. I fell in love with B lymphocytes when I was a PhD student. And I moved to Mill Hill in the National Institute for Medical Research to work with Mike Parhouse in the characterization of B cells, of molecules providing signals for B cells. And it was fashionable at that time to produce monoclonal antibodies. So we use that methodology to characterize several monoclonal antibodies recognized in B lymphocytes. And unfortunately, at that time, we didn't have the capacities in the lab to clone the molecules. Although we tried, we didn't succeed finding what was the antigen recognized by these monoclonal antibodies. But for that reason, I was very lucky to move to DNEX. DNEX at that time was the champion of cloning cytokines, cloning different molecules. So one of the antibodies that I produce induced the proliferation of B lymphocytes. So it was good for me to move there to characterize this antibody and to find that the antibody recognized a molecule that is now known being CD38. And part of my life developed in the characterization of CD38 on B cells. However, I have other antibodies, and one of these antibodies that we call R8, the antibody, when you insolubilize the antibody, for example, in a plastic plate or in the tissue culture plate, you can see that B cells, activated B cells, take this shape, this morphology, this quite strange morphology. It was an interesting observation. It was a curious observation. However, a part of these changes in the morphology, there was nothing interested to pursue the cloning of the molecule. So the molecule was then, the antibody was then put on the drawer in the refrigerator, and we forgot this antibody for several years. When I came down to Mexico to take a position in the institute where I'm working now, one of the things that I made was I was very curious about this antibody, was to use the antibody to clone the molecule. And with the help of Paul Kincaid, a very good friend of Mike, and then a very good friend of me, he provided me with some tools. Then I characterize the antibody, and the antibody recognizes CD44. And we published that long time ago. But a part of that is, I insist, there were nothing interested on that. Then we start looking at why the shape of these activated B lymphocytes changes. And this is a movie that we made with a very old VHS video camera on a very old microscope. And it would take a couple of minutes just to see how B cells move and how B cells make these dendrites, these structures on the membrane. And I was so shocked because the image that I have from lymphocytes is a round cell that probably was rolling on the tissues, but never made these fantastic movements. That it has that big motility. So at that time, after getting those results, we decided to study the cytoskeleton, because in order of the lymphocytes to take these shapes, to move that way, it has to produce different changes in the cytoskeleton. So we start the characterization of actin, the actin cytoskeleton, the tubulin. Those are B cells activated with cells with this spreading phenotype, and we see that the actin and tubulin, both of them, participate in order of getting this morphology. But another observation that was made at that time by my PhD student, Adriana Somoza, she used this drug, the butanedione monoxide, to treat B cells before spreading. And then we put the cells to do the spreading, and this is the vehicle, the MSO. And as you can see, the activated B lymphocytes put on top of anti-CD44 antibodies take these fantastic shapes that we have been observed all the time. However, when the cells were incubated 30 minutes before the spreading, all the cells remain rounded. They didn't change the morphology. So we were very intrigued with these results, and we started learning, looking for information about the role of this drug. And this drug induces or blocks the activation of the ATPases, and especially blocks the activity of different myosins. It's not a very specific drug. However, it gives us the idea that myosins and these motor proteins should participate in this phenomenon. So then we start looking for myosins on lymphocytes. Myosins, as I mentioned, are motor proteins that move along the actin filaments, and this motility depends on the hydrolysis of ATP. And we didn't find much information of myosins on immune cells. However, we look for members of the family. The family mostly studied are the class 2 myosins, that are the motor proteins. They are pretty similar to the myosin present in the muscle. However, there are some other members of different families of this protein. All of them contain a motor domain. This motor domain is contain the ATPase activity that allows these proteins to move along the actin filaments, but also they contain a tail domain, and tail domain are pretty different among the members of this family. So we concentrate on these two members of the family, the class 1 myosin 1, initially 1C and then 1G, and later on we start looking for myosin 1A and myosin 1F. But these two families of proteins are, as I mentioned, contain the motor domain, the neck domain, where calmodulin can bind and regulate the motor domain, the activity of the motor domain, and the TH1 domain. The TH1 domain contains inside a region that can bind to phospholipids in the membrane. So in a way, these proteins remain attached to the membrane, but then can move portions of the membrane thanks to the motor domain. And also the other class 1 myosins, the long tail class 1 myosins, contain also TH2 and TH3 domains. This TH2 is a protein-rich domain that allows these proteins to interact with some other proteins. And the TH3 domain is also a region that through the SH3 domain could bind to different proteins, different components of the proteins. So starting with the characterization of myosins, which myosins are expressed on B lymphocytes, as you can see here, it's RT-PCR or Western blot. So we can find myosin 1C, myosin 1A, and myosin 1G. We work mainly with these two, although we start working with myosin 1C. We concentrate in these two, especially because we were able to get the knockout mice from Steve Shaw in the NIH and from Richard Flavel from Harvard. And this is the expression of these proteins in B lymphocytes. So, as I mentioned, the cells, when a lymphocyte moves, it moves through the formation of different protrusions. The protrusions can be philopodia, and this philopodia is very rich in myosin, in actin and myosin, in order to promote the motility of the cells. It contains some adhesion components that allows later on the cells to form stress fibers through the attachment of these adhesion molecules to the substrate, and this allows the cell to move on. Myosins also have different participate in different mechanisms of the cells. For example, not only in motility but also in the movement of secretory granules, of phagocytosis, on the formation of synapses, and especially myosin 1C is known to work as part of the transcription machinery in different cells. So just to start with myosin 1G, as I already mentioned, during spreading, for example, B lymphocytes produce and move myosin 1G to the philopodia expressed on those cells. This is not the Giardia. This is a B lymphocytes moving all along in the media. You can find the accumulation of myosin 1G in all the philopodia on the cells with that colocalize with actin. So there is a very nice colocalization with cortical actin in all lymphocytes. We purify the pili, these structures, from B cells, and we found that myosin 1G was very rich there. Also, something that was already known, B cells are able to phagocytate molecules. For example, these B lymphocytes phagocytated latex particle, and we found that B cells can endocyte not only latex, but also can endocyte bacteria, for example. In this case, Staphylococcus aureus, we have used also E. coli, etc. And B cells can eat, not as efficient as a macrophage, but they can eat quite good. But one of the first surprises that we found is that the knockout mice, the knockout for myosin 1G, can eat faster and more efficiently. So I won't show you the experiments already. This information is already published, but we looking for an explanation of this difference. We found that myosin is very important in maintaining the tension of the membrane on the surface. So that means that the wild type has a very stiff tension on the membrane on the surface, but the knockout mice has less tension, so it's less stiff, this membrane, and that's why, for that reason, that B cells can eat more particles. But as I mentioned, myosin 1G can bind to the membrane in one side and to the cortical actin in the other side, and this mechanism is used by lymphocytes, for example, move different molecules, to move structures. One of these, for example, is CD44, as I already mentioned. CD44 was the trigger to start the analysis of myosins. So if we can induce capping, if we induce capping of CD44, wild type cells induce very nice capping in 30 minutes, while the knockout mice is not able to induce capping. Also, the expression of CD44 on the surface of knockout mouse is reduced. That means, or that suggests that somehow myosin 1G was involved in the recycling mechanisms of CD44 to the surface. These knockouts didn't have consequence on the synthesis of CD44. The synthesis is exactly the same, but the expression on the surface is quite different among these two cells. Another difference that we found that was very interesting is that in the lipid drafts, as you know, lipid drafts are structures of cholesterol-rich structures that are on the surface of many cells, many kind of cells. And B lymphocytes contain these lipid drafts on the surface. And if you compare wild type in blue, for example, with the knockout in red, you can see less lipid drafts on the surface. This experiment was important because it explained why CD44 is less abundant on the knockout mice, because CD44 is part of these lipid drafts. So if there is less lipid drafts on the membrane, there are less CD44, and not only CD44 but also some other molecules that I will show you later. If we treat, for example, wild type B cells with beta-methylcyclodextrin, this takes sequester the cholesterol, and in that case, wild type B cells looks like knockout the myosin 1G knockout mouse. And also, if we induce immunoprecipitation of myosin 1G, for example, we take caveolin, or just the opposite. If we take caveolin, we can get myosin 1G. We think that there is not direct link between myosin 1G and caveolin, but these molecules stay together through the lipid drafts, and the lipid drafts are probably the platform where those cells use to move together. And the motility of the cells is impaired in the knockout mice. And the explanation of this reduced motility probably is related to problems in the expression of the CXCR5 receptor for CXC12 and CXC13 chemotactic factor. But I think I'm going over my time, so I move faster. But I'm going to talk about now on some in vivo experiments with myosin 1E. Myosin 1E is as I mentioned, long-tail class 1 myosins. So we decided to analyze in the knockout mice how was the motility or the recruitment of the lymphocytes in the inguinal lymph node. And just to compare, this is the venules, the venules that are part of the inguinal lymph node, and you can see some cells attached to the endothelial cells. These white dots are B lymphocytes, and some of them are moving very slow and then faster in the circulation. But you can easily see that some are recruited and some other moves very slowly. But what happened in the knockout? If you compare that with the knockout, you can see that, first of all, there are less lymphocytes that are attached to the endothelium, and most of the cells are moving through these venules in the lymph node. So that means that we can see less recruitment of B lymphocytes on the lymph node in the knockout mice. So just to show you that we can see less recruitment, you can see less white dots on those venules in the knockout mice. And the explanation, well, apart the motility is reduced in the slow rolling mechanism. This slow rolling is mediated directly by interaction of LFA1 with ICANN1 or BL4 with fibronectin or other ligands. So that means that the expression of these molecules, LFA1, BL4, or CD44, are reduced on the knockout mice. And that's the reason why those cells, the knockout mice, move faster in the venules and are recruited, are less recruited in the venules on these mice. But just to show you a more evidence of this interaction, we look for the adhesion of B cells on fibronectin as a surrogate ligand for BL4, or hyaluronic acid as a surrogate ligand for CD44, and you can see less adhesion of those cells. Also, we can analyze the same. For example, instead of using these ligands, we use directly endothelial cells. You can see more cells bound to endothelial cells in the wild type, and less cells bound to endothelial cells in the knockout. So that means that those cells are the knockout myosin 1E cells are less active in binding, and also they are less active in producing these structures that, as I mentioned earlier, are part of the mechanism to induce the binding of those cells to the endothelium. Just quickly, just to show that this is the motility of wild type and knockout B cells upon activation of 612 chemokine. And the mechanism, we look for the mechanism. What is the mechanism that allows, for example, or that impairs knockout mice to bind specifically to LFA1? And we know that LFA1 is bound, is interacting with the focal adhesion kinase, and this focal adhesion kinase upon phosphorylation induce the activation of PH3 kinase that then induce the activation of AKT, RAC1, and active polymerization that induce migration. Just to make the story short, we found a reduced phosphorylation of the focal adhesion kinase in the knockout mice upon activation with a chemokine compared with the wild type. And something that was very interesting, we induced the immunoprecipitation of myosin 1E, and we took and we found a FAC associated with it, or the opposite, immunoprecipitation with FAC, and we found myosin 1E localized in the same precipitate. We also look for activation of AKT. So the phosphorylation of AKT is reduced on the knockout mice. The activated form of RAC1 is reduced on the knockout mice. And to evaluate the participation of PH3 kinase, we use this drug. And we can see that the production of these protrusions of the membrane are reduced also on those cells upon the use of this blocking agent. So just to make a story short, these protrusions are somehow produced by the participation of myosins that move all these molecules to the surface and to promote the adhesion of lymphocytes to the surface of the cells. And you can see some B cells with several flotations on the surface. My last slide, sorry, I went too fast. This is the work that was produced by José Luis Maravillas, and now is an independent PI in the Nutrition Institute. He started working with myosin 1G, and then later on, Orestes Ortega worked with also with myosin 1G. Later, David and Daniel worked with myosin 1E. And unfortunately, I didn't have the time to talk about the work with macrophages, with the myosin 1F knockout, and with gamma delta T cells that was produced by Irving very recently in my laboratory. Sorry for going too fast, but I hope you could get some of the message. And I acknowledge again WAL and the Brazilian Society of Immunology for this kind of invitation. Thank you.
Thank you, Leopoldo, very much for a fabulous presentation, really interesting. We do have a few questions here. I have one that was sent to me by André Souza from the Federal University of Bahia, and he is interested in learning whether the disruption in myosin will also affect antibody production later on by these cells?
No, unfortunately. Well, we look for changes, and I think changes in the production of antibodies, and we couldn't find any. And in fact, we tried to characterize, for example, the increase in susceptibility to some pathogens that we have on hand, and we couldn't find anything really relevant to report.
Okay, so Thiago Santos is also asking a question here. He's asking, would it be possible to modulate myosin in tumor cells or induce that B lymphocytes extend cytoplasmic projections over neoplastic cells in order to perhaps increase the internalization of tumor antigens?
This is the idea. In fact, one of the reasons that we have been analyzing cytoskeleton is because first of all, we have evidence that those structures are important for the contact of the cells with, for example, with T cells and with other cells. And as I mentioned, I fall in love with B cells, but I'm also working with T cells. And we are finding some very interesting results also on T lymphocytes. The reason that we focus on B cells is because I have more tools to analyze the physiology of those cells. But these structures, of course, are probably the same or quite similar in T cells, so it's worth to work with. And especially, there is very few people working on cytoskeleton and lymphocytes in general, or leukocytes in general. So it's a field that needs more people working in.
And do you know if there are differences in these dynamics, whether you look in B2 cells versus B1 B cells?
Yeah, there are some differences, especially because in order, for example, to see those differences, we need to activate B cells. And the B2 cells that we took from mice are usually resting B cells. So our animal house is quite good, and we get resting B cells. So we need to activate those cells in order to see all the changes that I already mentioned. But for example, B1 cells are already activated. Right. They are very active, and this is something that I would like to analyze more in detail, because, for example, the gamma delta T cells that we have been analyzing in the last 3 years in the laboratory are cells that are mainly found in the mucosa, and they are very active. and they are moved very fast. I'm really amazed how those cells move on the tissues, looking for antigen and probably working or participating in the regulation of inflammation on the sites. So yes, there are differences, and unfortunately, it's more difficult to work with those cells because it's easier to take the spleen than the peritoneal.
Right. Yes, exactly. Okay, so so thank you both very much. I think we are on our time. Thank you to all of you that came and watched the presentations and participated as well. Thank you, Emilio, for being here. There is one more question here by Patrícia Najib, who is a good friend from many years ago at she was at Federal University of Minas Gerais, now in Campinas. Patrícia is asking, sorry, Leopoldo, and yeah. I promise this is the last question. In obesity context, the myosin can be regulated by adipokines?
I don't know. The quick question is, I don't know. But that's possible. That's possible. In fact, there are many ways to activate lymphocytes. So I shouldn't be amazed to find activation.
Okay. All right. So thank you all so much. Thank you again, Leopoldo, and thank you again, Karina, for fabulous, excellent presentations. Many people here clapping and talking good about their images and slides. So that was really excellent. And I hope to see you guys again in a couple of months. We're going to have the next Immunolac in a couple of months, and you and your people from the group, so we can keep interacting together amongst ourselves. So that's.
Thank you very much, Wal, and thank you all of you. Thank you, Karina.
Thank you. Thank you as well, everybody.
Yes, great seeing you. Bye-bye. Thank you.